So, hello everyone. I'm sorry that I can't be there and participate in this exciting day-long discussion, but thank you for allowing me to present some thoughts via Skype. And I just want to apologize that since I'm not there, I may be making comments that are repetitive or you've already discussed, so I apologize in advance. What I'd like to spend the next few minutes doing is reviewing the criteria that were outlined by the 2017 International Study Committee for germline editing, and then layering on that some of my own personal thoughts and comments. So next slide. So this was a committee convened by the National Academy of Sciences and Medicine along with the Royal UK Society and actually even the Chinese Academy of Sciences, and it was a quite large committee that was international in its nature and spanned many different disciplines from scientists to lawyers to bioethicists to thought leaders from around the world. Of course, in no way could this committee completely include all relevant stakeholders, and I think that was an important part of some of the recommendations. Next slide, please. So out of this committee came a report. I will say that the task of the committee was to review the science, to assess the use of genome editing as a research tool in early human embryos and zygotes, its potential use in somatic cell editing, as well as in germline editing, and discuss the governance. So it was really quite a broad mandate, and as such, the sort of section on germline editing was probably not as thorough as it might have been, but again, the mandate was to be quite broad. So in the next slide, Barry Collar, who's the chief medical officer at Rockefeller University, in our deliberations, made a statement that what he felt like we needed to do was design some recommendations that supported the idea of generating healthy babies but not designer babies, and I paraphrase that to say that I think the goal is to generate healthy humans, not designer humans. So next slide. So I'm not going to review all the other aspects of this report, but instead focused on the criteria for heritable germline editing that the committee proposed. We recognize that, in fact, by saying that there might be a path forward in which heritable or germline editing might be considered, we were stepping over a line that had previously not been stepped over, at least by a sort of well-established and official body. So we recognize that this was a line that had not been proposed to be crossed. I will say, I think in retrospect, we as a committee made a mistake by saying that while we have outlined some criteria, the reality is, even as 2017 and even now in 2019, almost none of these criteria have been met. And I'm not going to read through them. I'm sure the audience there is very familiar with what these criteria were, but I want to break them out into two general categories. So next slide. One can say that one aspect of the criteria were sort of social and governance criteria. And this is not my area of expertise. Of course, I learned a lot by listening to people who are experts, and I know there are many people in the audience there who this is their area of expertise. So I'm not going to go into this in great detail, but my sort of lay summary of a lot of these criteria is that if one were to do germline editing, that it should not be done in secret, that it should be public, transparent, broadly discussed, and many of the criteria then were about how you would maintain that this wasn't done in secrecy while but also maintaining other things that we believe is valuable, such as patient autonomy and patient privacy, but without making this occur in secret. So what I want to focus on in the next few minutes in the next slide is the technical criteria that the committee proposed. which, as I mentioned before, are really quite strict. And I really want to focus on two technical criteria that were proposed. The first is, and actually was the number one criteria in the list, is that there has to be an absence of alternatives. And the alternatives, well, first of all, let me step back, is one of the assumptions of the whole thought process was that while there is debate about the rights of a couple for having a healthy genetically related child, I think that practically we know that there's such a drive for parents to have genetically related children. We assume that was a real driver, and we could not say that, well, there's no right to having a healthy genetically related child. So given that, then what we said is that, well, what if there are alternatives for a couple having a healthy genetically related child? Then there would be no reason for germline editing to occur. And so some of those alternatives, of course, include in vitro fertilization with pre-implantation genetic diagnosis. But I think what we have alternatives coming down the pike, and even is somatic cell gene therapy or even small molecule therapy that may transform a deadly disease or a lethal disease into one that's no longer deadly or lethal. And so what this means is that, well, I'll get to this later. My personal comment is that in sub-bullet point 2 there is people have argued, well, since IVF with PGD is inefficient, why can't one use that to increase the number of implantable embryos? And my feeling on that is that if IVF with PGD is inefficient, adding an additional manipulation to the process is unlikely to make it more efficient and generate more pregnancies. And the challenges for many of the examples in which you would want to increase the number of potential embryos to implant, you'd be exposing a large number of embryos to a process, and a large number of so-called healthy embryos or zygotes to a process that it didn't need to be exposed to. So you'd be exposing a zygote to reagents that were of no benefit to that potential human. So, you know, I think, and then the third bullet point there is to call out the UK health system who supports and pays for IVF with PGD for certain defined diseases and indications. And I think I find this a very appealing system because it allows parents to have a healthy genetically related child and thereby decreases the amount of suffering in the world. But it also has a side effect for many of these diseases of being economically cost effective. The other technical point I want to spend a little time on is the third major bullet point, which is the committee said that we should only edit 2 sequences that are common and known to be healthy in the human population. And I think this is a really under-recognized, very strict restriction on what can be done. So the idea here is that a pathologic sequence that's rare in the human population could be converted to a non-pathologic sequence that's common in the population. And exactly that sequence, not a sequence that is similar to or one could argue might be functionally identical, but what we're saying is the exact letters of the DNA have to be the exact letters that are found in a large number, the so-called what common means could be debated, but is common. And so that means that you wouldn't take healthy sequences and modify them to sequences that are bespoke or are not present in the human population. I will also say that, you know, people have made the argument that, well, there's a few human beings out there that have a sequence that I think might be beneficial. And my argument is that if there's only a few human beings out there with that sequence, we can't really know that in all environmental situations that a human might encounter in their life, it is truly healthy. It's sort of a argument that human evolution is a guide to what might be healthy or not in a broad range of humans. So on the next slide, just want to highlight that the work in China violated all of these criteria, or essentially all of these criteria, by secretly editing a healthy and normal gene into a novel sequence. And you can read all the ways I think it violated that. But I think the thing we as a community learned is that he was able to talk himself into that he wasn't violating these criteria. And so what that, I think, tells us is that if we just allow individuals in their own minds to decide whether they are meeting guidelines or criteria proposed, we are likely to get decisions that we would broadly disagree with. So a couple, next slide, sorry, is just wanted to bring up a couple more technical issues related to editing zygotes and specifically. So we, of course, because we don't want to generate new sequences in the human genome, we have to have highly specific processes to edit. And I actually think that with the current reagents, that is possible. That actually editing is now probably more specific than, say, meiotic recombination or the natural mutagenesis that occurs during gamete generation. The problem is that we would never be able to prove that in the zygote that was being implanted, because you would only sample one, say 10 to 20% of the zygote. You could never guarantee that the remaining cells didn't have an off-target mutation. The second major challenge is the issue of chimerism, and it goes to much the same argument that I made about specificity, which is that we know in genome editing that you have the risk of creating insertions and deletions on one allele. By definition, all insertions and deletions are essentially unnatural in the human population. And so how would you determine that the zygote does not have chimerism with novel sequences in it that could then be passed along to future generations? So again, this issue of chimerism, while people are improving on it, you could never prove that an individual zygote was not chimeric prior to the implantation. The third point I want to make is technically generating using genome editing to generate only common and healthy sequences remains very challenging. I just mentioned the idea that indels are not a natural, common, and healthy sequence, even if we think in the somatic cell they might have some therapeutic benefit. And even when we and others do homologous recombination-based editing, we often introduce silent mutations into the genome in order to enhance the efficiency of the recombination process. But again, by definition, these silent mutations are not common and actually can't, and therefore can't be known to be healthy, even if we think they might be. Which leads to point number 4, is that while we have focused a lot on zygote editing, because that is technically what is most accessible and what has been done in human zygotes and a large number of animals and generating pregnancies in animals, I feel like what these technical issues might actually be solved by editing cells that give rise to germ cells rather than the zygote themselves. And so the next slide is sort of it's a slide where I've tried to depict all the various stages one might use genome editing. Over on the right-hand side, in vivo or ex vivo use to modify somatic cells. People propose to use embryonic or in utero editing, where the risk of inadvertent germline editing, I think, is higher. But what I've boxed in red is the concept of editing either spermatogonial stem cells that will give rise to sperm, or in induced pluripotent cells that could either give rise to eggs or sperm, and that then these gametes derived from the precursor cell would be used to generate a zygote. So let me first say that publications for editing of mouse spermatogonial stem cells have been published using various nuclease platforms. Those edited spermatogonial stem cell clones have been isolated and purified. In the case of Cas9 based editing, have actually generated sperm that led to offspring. But this has not been done yet in human spermatogonial stem cells as far as I'm aware. Of course, editing in iPS cells has been done in human iPS cells, and of course clones can be identified and sequenced that would meet the criteria of having no off-target mutations and on-target changes that are common and healthy in the human population. And again, mice have been derived from eggs derived from iPS cells. So this, I think, would be a technical workaround to the idea of zygote, the problems with zygote editing, and yet would still end up with changes passed along to future generations. So next slide. So a couple additional thoughts is that there's no such thing as one human genome. In fact, we're all different, and actually almost every cell in our body is different. So I don't believe in a sort of sacrosanct human genome, although I think there's some commonality. The second point I want to make is if you do heritable or germline editing in one country, it affects all of humanity, because we are now part of a global village, and people throughout their lives travel throughout the world. And so therefore, the decision somebody makes in one country actually affects all of humanity. Which leads me to the next slide, which is my thoughts on how there might be a translational path forward. Now, clearly, what I'm not saying is that there should be a moratorium that's permanent. Instead, what I'm saying is that for rare couples who the only way for them to have a healthy genetically related child is to go down this path, I believe as a pediatrician who takes care of patients with very rare diseases that we should establish a ethical, technically safe, and transparent pathway to do this. And this is some of my thoughts on this. It would be that a large and diverse international committee is created to regulate germline and heritable editing to create pregnancies worldwide. I don't believe this committee should oversee somatic cell editing or editing in human zygotes for research purposes. I think that would create a scope that's too broad. But the committee would supplement but not replace existing regulatory processes and authorities. So, for example, in the FDA, in the U.S., the FDA would continue to regulate such processes, and in the United States, as you all know, it's currently illegal. So you would not usurp sovereignty from countries if they had more strict rules. The committee might adopt some strict criteria, such as what we proposed in the 2017 study committee. And again, I go back to this idea of restricting to editing to sequences that are common and healthy, because I believe it solves a lot of the issues that people have brought up. Like, for example, what about enhancement? Well, if a sequence is common in the population, it can't be an enhancement because most of us have it. It will protect us from hubris and thinking that we're smarter than human evolution. And it would protect us from leakage into applications that go beyond the prevention of disease. Moreover, it would not fundamentally alter human evolution or genetics. And one could imagine that, much like the UK has done with IVF and PGD, you could generate a list of sequences that were acceptable to edit to, and thereby say, well, if you're not editing to exactly these letters, it's not acceptable. Of course, such an international committee would have no statutory power, and it would only gain that by having countries pass laws saying that unless this committee approved the creation of a pregnancy, it would be illegal in that country. And then finally, the really important part would be that the committee would approve or not a protocol only after public disclosure of the preclinical data on safety, efficacy, and chimerism, the consent form, the clinical trial design, with time for public comment and public consideration of whether this would be something that should be approved. So next slide. Again, I've said many of these things already. I guess the only thing I want to end with is point number 6, which is while I have focused a lot on the technical issues of germline and heritable editing because that's closer to my area of expertise, the societal, ethical, and governance issues are at least as important, if not more important, than the technical issues I've reviewed. So with that, I'll stop and hopefully leave some time for discussion. And again, I'm sorry I can't be there to participate fully in the rest of the day. Again, thank you very much.